Hello, welcome to Sage Green, where we delve into the wonderful interdisciplinary field of bioengineering. Today, we'll explore how the body reacts to the implantation of a biomaterial. We'll cover cell adhesion, communication, biomaterial testing, and the effects in the body of the implantation, including the immune response. To investigate biomaterial interactions, we first need to understand cell adhesion. You might have been taught that tissue is made up of its constituent cells, but you might not have known that the mesh that holds the cells together makes up the majority of the mass of the tissues in the body. The extracellular matrix, ECM, is composed of fibrous proteins such as collagen and elastin, and a gel of proteoglycans, which are polysaccharide chains linked to proteins. An important protein in the ECM for cell adhesion is fibronectin. It has multiple binding sites as well as binding to the cell surface and adhesion between the cell and ECM. Cells also adhere to each other and to the biomaterial where the ECM would have been if it is present. Zooming in even further, this is done in cell junctions with cell adhesion molecules, or CAMs, due to the range of proteins involved and type of junction connections using the ECM and another structural element, the cytoskeleton. There are a variety of ways a biomaterial can adhere to living cells. This can be used to our advantage by engineering materials to express certain adhesion. For example, a wide array of research has been conducted into regenerative materials to promote adhesive properties. Mitchell and Lowe reviewed the latest strategy of using small molecules to coat materials in 2023, as opposed to natural alternatives that come with more biocompatibility issues. This is when another important element comes into play, the cytoskeleton. The cytoskeleton is a complex network of protein filaments inside the cell that provide structural support facilitates cellular movement. Actin filaments, microtubules, and intermediate filaments are key elements of the cytoskeleton. During cell adhesion, these elements undergo dynamic rearrangements influencing the cell's shape and behavior, aiding in cell adhesion among other cell processes. Another important cell process that impacts biomaterial interaction is cell communication. Cells signal by excreting a molecule from one cell to another, whose signal is then received and transduced throughout the receiving cell, leading to an eventual response. When designing a biomaterial, you first need to take into consideration the molecules that are going to be excreted when they come in contact with it, as they have different potencies and lifespans. Additionally, there are also different methods of cell signaling in relation to the cell that needs to be accounted for. After the molecule has been secreted, it can bind to the receptor on the plasma membrane, which can be recognized by either ions, enzymes, or molecules that can bind to a specific protein. This is followed by a pathway leading to an eventual cellular response, in which proteins are often turned on or off. Cells are also able to signal more precisely and efficiently by combining signaling molecules, using one molecule to signal to many receptors, and by changing receptor expression. There are many different signaling molecules, but the most common ones and the most important for biomaterial interaction are cytokines, and more specifically interleukins. Cytokines are proteins and signaling molecules for the immune system, and interleukins are a class of cytokines that mediate communication between leukocytes. Their role in the immune response is vital to understanding transport rejection. For example, cytokines are released to activate major histocompatibility complex or MHC molecules that in turn express intracellular antigens to notice and destroy foreign cells, which is useful for when we get infected, but not necessarily when we get a transplant. Cytokines also regulate inflammation and immune system cell differentiation, among other functions within homeostasis. Because of their role in the immune system, and inflammation, hypersensitivity, and overexpression can lead to conditions like autoimmune disorder, rheumatoid arthritis, and cancer. However, our understanding of these signaling molecules can also allow us to creatively solve these conditions and maybe reduce the chance of transplant rejection. For example, targeting specific enzymatic pathways to treat the inflammatory symptoms of rheumatoid arthritis, as written in a report in 2023. Because of complex cellular processes, including cell adhesion and communication, care must be put into testing a biomaterial before clinical use. Biocompatibility is tested first via risk assessment, then an in vivo test, and first an in vitro test, and then an in vivo test. During the risk assessment, The factors that influence toxicity are taken into account. The specific dose, duration, frequency, and exposure make negative effects range vastly. For biomaterials, the material itself, the site of application, and its surrounding biology also have an effect. More specifically, xenobiotics, foreign molecules, can have chemical, biological, and physical effects on the surrounding cells. Even further, xenobiotics from implants can have systemic effects, which are those that travel to the rest of the body. For example, if a material is in contact with the brain, it can affect the whole nervous system. In vitro tests for biocompatibility are regulated under ISO and designed to test for negative effects on cell function, and can also test for toxicity linked to gene mutation if necessary. For example, in the MTT assay, cells' metabolic activity is measured after direct contact with the material. This is done by mixing MTT salt with cells that have been left to multiply on materials in an assay structure. Metabolic activity can then be calculated with a spectrograph, as the cells that have active mitochondria will convert the salt to a purple color. There are many other in vitro tests that can be used based on the application of the material. In vivo tests are then carried out often in small animals. For example, the intercutaneous irritation test tests for symptoms of inflammation after the injection of the molecules. As more innovative synthetic materials are being researched right now, there are challenges in studying long-term effects as well as obtaining accurate results for reducing animal testing. But right now, novel methods are being researched, for example, 3D tissue scaffolds to test drugs and implants. To help understand research into biocompatibility testing, it is also useful to delve into the adverse effects that biomaterial implantation can have on surrounding cells. Toxicity can be caused by materials in many different ways, leading to different cellular responses. Materials can be degraded within the body during long-term use and can leach into other parts of the body, leading to a variety of systemic effects. Different materials also have varying effects. Polymers can have chemicals left over from the manufacturing process, and metals can give off metal ions. These effects are caused by the chemical makeup of materials, but they can also be caused or exacerbated by the biological properties, such as the immune response and inflammation of the cells surrounding the implants, as well as the physical structure. This leads to research into potential carcinogenic and genotoxic effects of biomaterials. Biomaterials should be tested for mutations of genes and chromosomes with in vivo tests, like for cytotoxicity. They should be tested first with in vitro tests, like for cytotoxicity, and if necessary, in vivo tests later. In vitro tests can be carried out with bacteria to test mutagenic properties of genes, and mammal cells can be used to test for chromosomal or gene properties later. However, even with potential cancerous side effects, biomaterials can be used in the treatment of cancer for drug delivery, tissue reconstruction after surgery, and modeling cancer growth more accurately. The similarity of stem cells to cancerous cells also has the potential to be exploited, though the research is still very new. An important challenge right now includes using stem cells to regenerate tissue within the body, because unmonitored stem cells can very easily migrate and cause tumors, but research is promising. Another big concern with implanting biomaterials is the immune system and the immune response to them. Once surrounded by host cells, a foreign body is greeted with the innate and the adaptive immune system. Complement is part of the innate immune system that we are born with. It is a cascade of proteins that activate each other and lead to immune system functions, such as lysing and digesting of microbes. The final few properties that are actively activated eventually form a membrane attack complex, where they combine to pierce through the plasma membrane of the microbe, which lets water in through osmosis until the cell bursts. There are different ways the final steps can be activated, but the one most important for biomaterial interaction is called the alternative pathway, This is a spontaneous cellular action that is happening at all times in case the immune system has not previously encountered a particular disease or is unaware that the cell's antigens are otherwise foreign. It works randomly by binding to a cell surface, and if there are no biological markers that are abundant on host cells, for example factor H, it triggers the complement cascade. Macrophages play a big role in the innate immune system and are most famous for phagocytosis. They can detect a number of molecules often found on the surface of pathogens. Interestingly, they can also detect once a host cell has been damaged and needs to be destroyed, which can also be triggered by the surrounding tissue of biomaterials when they're implanted. This then leads to the foreign body response, in which biomaterials are degraded as damaged proteins affect macrophages. The macrophages can aggregate into a large lump of cells with blood supplies formed around the biomaterial, causing inflammation and rejecting the biomaterial. Materials that come into contact with the blood can also have specific negative interactions. Blood flow can be slowed or even stopped, leading to stress on the blood vessels and clotting. Inflammation can also be induced, leading to complement activation and attraction of pro-inflammatory macrophages from cytokine signaling, as you remember. Luckily, biomaterials can be fine-tuned, for example, to not affect protein absorption and therefore not signaling for inflammation. Other than reducing inflammation, it can also be embraced and used, for example, to make vaccines more effective at causing urgent and efficient response from the immune system. Transplant rejection can also have a similar process to autoimmune disease, which you may know as the body's response to host cells as foreign. This is an example of immune hypersensitivity. You might remember that MHC cells, mediated by cytokines, interact with T cells that lead to pathogen destruction. Since the exact makeup of the MHC is unique to each person, if non-self MHC is detected, this also activates an immune response. This can be alleviated by matching MHC, but is an imperfect solution. Now, some research is being focused on improving MHC analysis to better match patients using more advanced methods, such as looking at antibiotic patterns and the genes themselves that express this. And that brings me to the end of this video. I hope you joined learning about how biomaterials interact with the human body and how bioengineering can be used not only to better understand fundamental biological processes, but also pave the way for innovative solutions to address health challenges and enhance the field of medical technologies. Thank you for watching. I hope you feel as inspired by my biomaterials as I am. Goodbye.